So it's all been said already, so I don't know where to get started. But okay, my name is Wojciech. I work at the OTAT. And then let me get started. That's something that I was asked to, to show you. The objectives of my talk is to explain the importance of the immunogenicity drug during development and licensure, identify steps in the immune response to therapeutic proteins, compute the predictive power in silico, in vitro, and ex vivo methods used to evaluate individual steps in the immune response, distinguish between the validating a specific method and predicting clinical immunogenicity, and then estimate immunogenicity risk by determining the probability of anti-drug development and the consequences if such antibodies are generated. Why it is so important, that already has been covered. It compromises the safety and efficacy of the protein therapeutics. The human economics costs of immunogenicity to patients, caregivers, and the healthcare system are considerable. And then immunogenicity asks to the risk and cost associated with drug development. So immunogenicity made it to the headline of the New York Times back in 2002. Here's the quote from this article. Although the Eprex case is most serious, virtually all biotechnology drugs provoke immune response in some patients, though usually just tiny fractions. The reactions are becoming of greater concern as the number of protein drug increases. And after 10 years, nothing has changed, actually. The Novo Nordisk ran into this issue of one of the drug developing one of the drug had to be withdrawn with the clinical trials. And so after 10 years, we still have the same problem. Now, nothing has been changed in terms of the immunogenicity issue. However, something else has changed. So one can see significant increase in revenue generated by 10 top treatments in the last 10 years. Back in 2002, biotech products accounted for only 7% of revenue generated by the 10 top treatments worldwide. But that number went up to about 70% in 2012. And over the next years, we expect to have even more biologics. So it's very likely that there will be more issues related to the immunogenicity. It is very important. Therefore, there's so many different guidelines by EMA, FDA, WHO, and several white papers. But I'm not going to deliberate about this. Now, let's discuss briefly, because this also has been covered by the previous speakers. Let's discuss how we can experimentally assess immune response. So we can do the proteolytic processing. Not really. It is in silico prediction is very poor. We don't know unless we execute the MAPS assay. We can do the pretty good predictions in silico and good experimental assessment of the peptide binding to the MHC or HLA, if you will. We can do pretty much good work on peptide MHC2 presentation also by MAPS assay. In silico prediction of the T-cell recognition by TCR or the HLA peptide complex recognition by TCR it cannot be very well done experimentally or in silico. And then also we have some methodology to do the pretty good work on the T-cell proliferation. Now, this is one of the papers published in our lab previously, long time ago, about what are the pharmacogenetics of immunogenicity. So you have 4 cases. You might have the self-peptide or the foreign peptides. One can be low affinity, one can be high affinity. So this is very obvious. If you have the low affinity self, there will be no peptide binding to CR. In the same case will be for the low affinity foreign peptide. If you have the high self-affinity peptide, because this epitope has been removed, there will be no TCR engagement. And therefore, the ADA will be unlikely to develop. However, if you have the high affinity foreign peptide, there will be TCR will be engaged, and then ADAs could develop very likely. So now, in this case, so there are some patients, like I'm talking here about the hemophilia 8 patients because I work in the division of hemostasis, and we are very interested in this. and the factor VIII was always problematic protein. So now, this was paper published back in 2012, and here you can see that there are patients that have different level of protein foreignness, and then it has been shown that you would have a higher risk of getting anti-drug antibodies if you are on the right-hand side. That means you have the large deletions or multiple exons or single exon deletion or nonsense mutation, and then, or if you have the less of native protein, that means higher truncation, the higher chances of developing anti-drug antibodies. Now, let's summarize very quickly the mismatch and the peptide HLA affinity as immunogenicity biomarkers. Yes, they're pretty good predictors. So here you're looking at the AUC, so that means area under curve, and then where you compare the true positive rate against the false positive rate. If you take into account the sequence mismatch, you're going to get AUC of about 0.72. I have to just mention that the AUC of 0.5 is, there is no match, and AUC of 1.0 is very good correlation. Then if you will do the HLA typing, then you will shift your AUC to about 0.9. So that means that this is pretty good predictor of the immunogenicity. Now, the problem is also that oftentimes we don't have the HLA information from the patients that are, that are being given the new biologics that are the part of the clinical trials. And that has been an issue previously because we couldn't simply do it because it would be too expensive. So it all has started back in 2001 when the first human genome, for the first time the human genome was sequenced. So the total cost was about $2.7 billion. That number went down in about 2007 to about $1 million. Now, 2013, we could get the human genome sequence for about the cost of 2,000 Honda Accord. And right now, it is really available. The genome cost of genome sequencing can be done for about the price of $1,000. And then it is predicted that in a couple of years, that might be a regular medical procedure. Here is how our lab is going about the immunogenicity predictions. So you have on the left-hand side, you have, we call it a heat map, and then in this heat map, each row corresponds to one HLA allele, and each column corresponds to one length 15 mer peptide centered at any given amino acid. And then each square on this heat map is colored according to the binding affinity between the HLA and the peptide, right? So this is very simple, and then we can actually, do whatever we want here. We can set the different cutoffs, and then we can see whether, at least in silico, this peptide will be immunogenic or not. Now, we don't like this way to look at this data, so we came up with sort of a promiscuity score. So in this case, what you can see, you can actually generate this graph. You can actually generate this graph using the data from the graph on the left-hand side. So this will take into consideration also the frequency of any given allele in the population. But better yet, what we like to do, we like to show like this sort of a bar graph, because in this case, the dash in the middle tells you what is the expected chance of binding between the HLA and the 15 mers to any given population. So now I'm going to be referring later on to this kind of promiscuity scores. So now, is this promiscuity score good predictor of the clinical outcome, and if this promiscuity score can be correlated to the clinical outcomes? And again, this paper shows that yes. So if you have the prevalence of the non-anti-drug antibodies. So actually the peptides, like this is again the example of the factor 8 studies, and it shows that the factor 8 peptides from these regions, especially the regions with those mutation, bind more to the MHC2 variants. So I guess yes, it can to some extent. Now, as already mentioned by the previous speaker, that there is another types of assay called MAPS, where you can actually combine the proteolytic processing with the peptide binding, and then you can directly measure the complexes that are exposed on the cell surface. And I'm not going to go into details probably here, but this is how this assay works. You are doing the LS mass spectroscopy analysis and identifying the peptides that are found to be exposed to the T-cell receptors. Now, you've seen this before. Again, this was the work that we've been doing for the last year or so, and then again, as Jeffrey explained, it was 2 independent studies, and then both studies show that the peptide derived from the tetraptacok alpha molecule has been exposed to the T-cell receptor. So that means actually that there is pretty good correlation in this case between the MAPS assay and the clinical studies outcomes. Sort of, we're very interested in this also, the peptide MHC T-cell receptor binding. However, I mean, there's no really good experimental method to do that. You know, the reason why is that there is a large collection of gene segments that encode the variable regions of TCR sequences, and these genetic fragments undergo the arrangement. Thus, each T cell possesses the unique T cell receptors. There is an enormous number of these molecules. There is 10 to 18 different T cell receptors. The vast diversity of these TCRs enables the immune system to recognize a wide range of potential pathogenic threats, and the vast diversity of TCRs makes it difficult to determine the antigen specificity of TCR from its sequence. And so what is this number? This is a million of trillions, or if you, it could be like 30 million times longer than humans have been on Earth, or million times the information on the internet, or simply all the grains of sand on the Earth. However, there's some hope. So there are 2 recent papers published in Nature that show that there's some algorithm that you could use to predict the antigen bindings specificity of novel T-cell receptors. So there's one paper, they reached about 80 to 90% accuracy in predicting the antigen binding to the T-cell receptor. And the other paper, they have actually did the 10 synthetic TCRs, and they predicted to be specific for M tuberculosis antigen, and 8 out of them when demonstrated to bind the antigen. So this method becomes better and better, I guess, and we'll be able soon to use this as another sort of confirmation test in our immunogenicity assessment. Okay, about bioengineering molecules, why I have to go back there again. Quote from the guidance, bioengineered proteins involve the introduction of sequences not found in nature and may contain new epitopes, and this could elicit immune responses. The reason I'm bringing this one up again, because we are trying actually to deimmunize this analog of factor 7A that has been made by Novo Nordisk and failed during the clinical trials. So again, as Jeff already mentioned, there was only 3 substitution, D, V, and Q. And then unlike for the wild-type factor 7A called Novo7, where the incidence was pretty much 0, the incidence for the tetraptacok alpha was about 11%. Again, the company withdrew this molecule back in 2012. And then, unfortunately, only after phase 3 clinical trials, they spent so much money on this. But however, for us, it was good because that provided for us a novel model to evaluate the usefulness of all these non-clinical immunogenicity assessments. why this is so important not to have the antibodies for factor 7, unlike for factor 8? So the hemophiliacs are, the hemophilia is bleeding disorder caused by lack or defective of factor 8 or factor 9 proteins. Cannot be cured, but can be nicely managed right now. The most significant impediment to the modern management of hemophilia A is the development of anti-factor 8 antibodies. It's about 30%. And this and then in case of anti-factor 8 antibodies, so-called bypass therapy is used, which includes infusion of recombinant factor 7. Treatment is extremely expensive. Again, why they cannot be, why they have to be 0% incidence for these antibodies? Because simply these patients are not deficient in factor 7. So you can imagine if you have the factor 7 antibodies, they could cross-react with your endogenous protein, and that could result in serious life-threatening conditions. So what Novo Nordisk did was they actually developed the molecule that's supposed to have, I'm not going to talk about the efficacy, however, they claim that it has the better efficacy, and it has the very similar safety profile. So, and then once the clinical trials were, once the molecule was withdrawn and all this data revealed, we are able actually to identify all these patients in red have developed the anti-drug antibodies, right? So now we didn't know this only until all this data have been published by Novo Nordisk. Now, so what we have to consider the potential T-cell epitopes and then what could be the consequences. So this is the summary of the first part of our study. So we have used several assays in silico binding affinity. So this assay, using this assay, we could identify that one of two mutant peptide binds with high affinity to several MHC2 class variants. By using in vitro binding affinity, we confirmed in silico findings. The question was, is the streptococcus alpha processed and processed and are highly affinity mutant peptides presented on the MHC class 2 molecules? Using the MAPS assay, we're able to confirm this one. The foreign peptides that bind with the high affinity elicit T-cell responses, yes. And HLA typing of patients. So now the conclusion was that all ADA positive patients carry at least one MHC2 that binds to mutant peptide with high affinity. Okay, so here's the first story. So if we knew this information, we in principle should be able to exclude those patients from the trials, right? So this study is done. Now, what we're interested in right now is what to do or how to change this molecule so all patients can equally benefit. So we simply have to sort of re-engineer this molecule. And so the first approach was very simple. We're using only one-step approach. So we wrote the software in Python called simulated annealing, where we would fit our sequence and then randomly substitute the amino acids, generate the new sequence, compute the affinity, and then here was the input into the software, and here was the output into the software. So if we generated something that had the significantly less, showed significantly less affinity, we would consider this one as to be deimmunized. Now, we decided to select one of the alleles that was found in the patients during the clinical trials, and then do the binding assay using the purified HLA. And then when you compare the streptococcus alpha binding affinity to the 1101 and then to deimmunized molecule, you see the significant difference. The problem with this molecule is that like whatever we made was had no enzymatic activity. So now, why that was the case? This is fairly complicated molecule. You can see here this is a heavy chain. There is a light chain. This structure is in the presence of the tissue factor. You can see the catalytic site and the active site here. And this is exactly the same view, 180 degrees. And then there is a blowout sort of from this region shown in cartoon. And you can see that really what happens here is not very well known, because there is like allosteric activation of the active site by the tissue factor. So basically what Novo Nordisk has done, they have, by introducing this triple mutation, they have mimicked the binding of the tissue factor to factor 7. So now our goal is actually to not only change this active site, but retain its intrinsic activity. So we develop a new algorithm. So now this algorithm sort of considered 3 different steps. So the step number 1 is just simply promiscuity score. So on this map, you can see actually the 2 different colors. White, it is not. So we call it not allowed, and this is allowed. So whatever is left on the heat map, you should in principle be able to change and retain and then decrease the binding between this particular sequence and the HLA. The second step was to include the conservation, so throughout all these species, different species. So we ended up having only this green fragment here. And then, fortunately, there was one paper published by the group of Professor Marks from UC of Harvard, where she has used this so-called epistatic model that allows for predicting the effect of mutation that explicitly captures residues dependencies between positions. So it's also through space. So then once we apply this protocol, we ended up having only 1, 2, 3, 4, 5 allowed position. Now, first of all, this is a very important graph. This is the Thrombin Generation assay graph that we always use to measure the activity of factor 7. And you can see different constructs. So the positions here corresponds to the number of amino acids on factor 7A. And then here you have in green, you have the Novo7 molecule. So this is the activity of Novo7. And it's important to explain that the Receptor alpha, why it was so much better than the factor 7, because its intrinsic activity was much higher. And all those experiments were done in the absence of tissue factor. That's also important. So basically that confirms that this triple mutation will mimic the binding of the tissue factor, the factor 7. So okay, so now, too fast. So now what we're able to do. So basically what we're looking for, we're trying to validate the red dots. So then we couldn't choose 30 different designs, 4 different designs, because there's a lab that consists of, there's only 5 people and only I'm making this stuff. The other people are theoreticians. So we're able to manage this. We purified this protein, we couldn't activate it. And that was confirmed by the Thrombin Generation. So we basically killed the molecule. Even though it's deimmunized, in theory at least, it's dead. So then what we did, we have chosen the other red square. And then in that case, this is non-reducing, this is reducing. We're able actually to make the molecule that is deimmunized. So this is the quadruple mutant right now. And then it's deimmunized at the same time. It retains its intrinsic enzymatic activity. So now we're trying to understand why that's the case, and then we we're trying to correlate our algorithm structure and then activity of the molecule. So now you can see again Novo7 activity, Receptor activity, and then you have randomly chosen residue that is not allowed in our algorithm, right? So in that case, it is dead, it is buried, it's not exposed, so that makes sense. Position number 301, red square, exposed. Okay, now we got a little bit confused about this position because it was shown not to be allowed. However, it was partially exposed. This is some sort of intermediate state between the Novo7 and the factor 7A. So now, but what about the immunogenicity? So we have run the internal controls in the lab using only one patient's cells. It is very promising. And right now we're making the proteins, and we will hopefully very soon. The ELISpot won't be an issue, but the MAPS, where you need about 25 to 40 milligrams of protein, might be an issue. However, we're going to be using the ELISpot using the cohort of 50 donors, and then we will make, using the full-length protein, our demonized variant will run the MAPS assay to demonstrate the new epitopes is not presented by the donor HLA molecule. But the most important thing is, like, what we're really trying to understand is we're trying to see if those 4 pieces correlate, right? And how well. And I think this is the excellent example because you have done something. So you went through the clinical trials, you have done all this analysis, and then you have another molecule that you can directly side by side compare with this that failed in the clinical trials. So that will be a very good, whatever we're going to get will be beneficial. Whether this is going to be demonized or not, we'll know how well those tools are now, what is the predictive value of these tools. Now, how do we use these results, the results of these methods to make drug development or regulatory decisions? So those are the products that we regulate actually, and then you have the Omontis, and here it increases as the probability of developing anti-drug antibodies increases. So Omontis has the lowest probability, followed by the Receptor alpha, and then factor 8, I'm sorry. Now, what are the consequences if anti-drug antibody or neutralizing anti-drug antibodies are generated? So now let's start from the alternatives. Is there any alternative to factor 8? No. Any alternative with Receptor alpha? Yes, you have the Novo7. Any alternative to the Omontis? Yeah, you also have, right? What is the immune adverse event? You might have anti-drug antibodies for both factor 8 and factor 7. However, you might have anaphylaxis for the Omontis. Now, is there any bypass or salvage therapy? Factor 8, yes. Factor 7, no. Omontis, no. Consequences, you can clinically manage this. This can be life-threatening, especially if you develop neutralizing antibodies. They can destroy your endogenous protein. And then in case on the Omontis, they can be very severe, leading even to death. So now, what would be your decision? In case of the factor 8, you can market it. In case of the Receptor alpha, you have to discontinue. And because there was very low probability of anti-drug antibodies at the very beginning for Omontis, they were allowed to market it. However, then that was followed by the withdrawn from the market after the antibodies development. In summary, immunogenicity is a significant impediment to development of therapeutic proteins. Decisions about the progressing with drug development and licensure when immune response are detected generally follow a risk-based strategy. Many therapeutic proteins provide the only medical option in otherwise life-threatening illnesses, mitigation strategies for immunogenicity. Emerging repertoire of computational experimental tools offer a comprehensive and increasingly accurate estimation of immunogenic potential. Concurrently, the development of mathematical models of immunogenicity offer a potential platform for synthesizing data from diverse set of assays and platforms. The utility of non-clinical studies and the willingness of the wider community, that's very important, to further investigate this will depend in large part on establishing clear association with clinical outcomes. And with that, I would like to thank you so much for your attention. Thank you.
Thank you very much. Okay, questions? Nobody wants to ask.
after you made your deimmunized versions, you go back in one slide where you showed that there were 2 versions with lower immunogenicity, but it was what test did you do for that?
Which slide?
A couple
We did
of slides.
we did the ELISpot
ELISpot.
and the
Okay.
binding assay.
Okay.
But the maps would be something that we would be really interested in,
Okay. Thank
because that will use the full-length protein.
It's not something you've shown, but I've heard the story being presented, and one of the things that was puzzling at the time it was presented is for the story of the recombinant factor 7A is when you're trying to confirm the MAPS results and find a peptide that elicits T-cell response, the size of the peptides was critical.
Mm-hmm.
Did you have additional explanation to that?
No, except for that the peptides ranges anything between 15 and 25 amino acids.
I've heard only the long peptide were working. The short peptide that contained the core.
I mean, you
were not really
cannot decide.
T-cells.
You cannot decide on. Oh well, you're not talking about the MAPS assay. You're talking
No, I'm
about
talking about the T-cell assay
Right.
that were done with
Yes, the
overlapping
15 didn't work, and
Yeah.
then 20 tumors.
Yeah, yeah. That
No idea why the 15 didn't.
Okay, no idea. Okay.
No, I guess it wouldn't bind properly because it was too short. Or right now we're using actually 25th MERS.
Wow.
Yeah, and they look very good, and the 15 MERS were not successful.
Okay, because most people use 15 MERS, so okay.
Yeah, just to comment on Laurent's comment, we started this discussion over breakfast, right, with the length of the peptides. I suppose one thing we're looking at for the MAPS type pro-present results is, about the peptide length and, also what you sort of see in amino acid presence, and distribution outside the binding group. So, there's the possibility, of course, for those amino acids, they probably don't interact with MHC, or they might, I don't know, but they might interact with the T-cell receptor and wrap around it, who knows. Presumably, once that's further out, it'll thermodynamically contribute less to affinity, but you might still get enough of a kick to make a difference from, what's beyond 15, who knows. But
No, I understand, but how do you imagine that the peptide will be exposed to the T-cell receptor without being bound to the HLA?
No, it would be. I mean, whilst it's bound to the HLA,
Oh, right.
it could bind slightly differently to the TCR on the longer variant. So that's possible, I suppose.
I guess there are certain examples where peptides can loop around on the HLA binding group, so there could be some evidence of that as well.
Yeah, I don't know. This has to be, finished, and then hopefully we'll know a little bit more about this. But we can let it go like this because it's too much has been done there, and then we'll see.
Great. Well, thank you very much.
Thank you so much.